Okay, so welcome back to this next video in which we are discussing the RAS-RAF MEK ERK pathway. Okay, so we've discussed now the activation of our generic receptor tyrosine kinase by its ligand, and as an example, of course, we're using the epidermal growth factor receptor with the ligand epidermal growth factor. Okay, and what then happens is once the ligand binds to the ligand binding domain of the receptor tyrosine kinase, this causes a change in conformation in the ligand binding domain that then allows it to dimerize with the ligand binding domain of another receptor tyrosine kinase that has also got ligand bound to its ligand binding domain. Okay, this then pulls the two tyrosine kinase domains on the cytoplasmic tails of these receptor tyrosine kinases which have dimerized very close together. Okay, and what now happens is a process known as trans-autophosphorylation, where the tyrosine kinase domain on one of these receptor tyrosine kinases is going to phosphorylate the tyrosine kinase domain on the other firstly, okay, and then the other one will reciprocate and phosphorylate the tyrosine kinase domain on the first. Okay, and the phosphorylation of these tyrosine residues within the tyrosine kinase domains of these receptor tyrosine kinases is going to serve to activate the tyrosine kinase domains. Okay, so previously they were extremely inactive, but now they're extremely active. And what they're then going to do is phosphorylate tyrosine residues that are outside of the tyrosine kinase domain of each other. Okay, so the tyrosine kinase domain of receptor tyrosine kinase number 1 will phosphorylate tyrosine residues anywhere within the cytoplasmic tail, basically, okay, of receptor tyrosine kinase number 2, and then receptor tyrosine kinase number 2 tyrosine kinase domain will phosphorylate the tyrosine residues in the cytoplasmic tail of receptor tyrosine kinase number 1. Okay, so we've now got a large number of these phosphotyrosine residues that are on the cytoplasmic tail of our receptor tyrosine kinases. We want to see how are these going to activate the RAS-RAF MEK ERK pathway. Okay, so let's go over the page and see this. So, the first protein that is going to bind to one of these phosphotyrosine residues on the intracellular aspect of these receptor tyrosine kinase dimers. Okay, so let's just redraw our picture out here. So once again, here is the amino terminus of our receptor tyrosine kinase. Here is the membrane spanning alpha helix. Okay, then we have our tyrosine kinase domain on the cytoplasmic side, and then we have the C-terminus over here. And we've got our ligand bound there. Now it's dimerized with another receptor tyrosine kinase here, which has also got ligand bound to its ligand binding domain. And now we've got the membrane spanning alpha helix. And remember, usually these two receptor tyrosine kinases will be the same receptor tyrosine kinase. Strictly speaking, they can be different as long as they're in the same family. Okay, so remember I told you that there are 58 different receptor tyrosine kinases in humans. Okay, and these are grouped into 20 families. If your receptor tyrosine kinase is in the same family as, well, basically, they can dimerize where one is not the same as the other, providing that the second one is in the same family as the first. Okay, so for instance, the epidermal growth factor is the one that we have been studying. This can dimerize, for instance, with other members of its family. Okay, so remember I told you that the epidermal growth factor receptor is also called ERB1 or HER2, standing for human epidermal growth factor receptor 1. Okay, there are other members of the ERB family. So for instance, there's ERB2 is another member of that family. Okay, there's also ERB3 and ERB4 as, to go further. Okay, now ERB2 is quite a famous receptor. It's called HER2. It's the target of the drug trastuzumab and is upregulated in breast cancer. Okay, well, many forms of breast cancer anyway. So basically, just to illustrate this principle, they don't necessarily have to be the same receptor tyrosine kinase, the two that dimerize here, providing that the two receptor tyrosine kinases are in the same family. Okay, they can then dimerize. So for instance, these two are both in a family of receptor tyrosine kinases known as the ERB B family. Okay, so they can dimerize, but you can't, for instance, get the epidermal growth factor receptor dimerizing with the vascular endothelial growth factor receptor, which is another receptor tyrosine kinase, because those two are not in the same family. Okay, right. So they have to have similar enough ligand binding domains that they can dimerize. But we'll keep this simple and think just about having two identical receptor tyrosine kinases dimerized together here. Okay, so we're imagining two epidermal growth factor receptors dimerized together. Okay, then we've got our tyrosine kinase domains down here, and I've realized that I'm gonna, well, I can get around that, I suppose. Okay, so let's colour in the tyrosine kinase domains once again in blue here. Okay, and we'll colour in the membrane spanning alpha helices in red. So here's one of the first receptor tyrosine kinase, and here's the second one of the second receptor tyrosine kinase. And the two tyrosine kinase domains have phosphorylated each other and are therefore now active. And once they have become active, they've then phosphorylated tyrosine residues all over the place, basically, in the cytoplasmic tail of their partner. Okay, so receptor tyrosine kinase number 1's tyrosine kinase domain has phosphorylated tyrosine residues in the cytoplasmic tail of receptor tyrosine kinase number 2, and the tyrosine kinase domain of receptor tyrosine kinase number 2 has phosphorylated tyrosine residues within the cytoplasmic tail of receptor tyrosine kinase number 1. Okay, so we now have these phosphotyrosine residues which are going to serve as docking stations for other proteins. Now, the protein we're going to be interested in, since we're looking at the RAS, RAF, MEK, ERK pathway, is a protein known as GRB2, but there are many, many different proteins which are capable of binding to these phosphotyrosine residues on receptor tyrosine kinases. Okay, so this protein here is going to be called GRB2. Now, what does this stand for? This stands for growth factor receptor binding protein 2. Okay, so the G is for growth factor, the R is for receptor, okay, because many of the receptor tyrosine kinases are growth factor receptors, okay, and then the B is for binding protein, okay, and I'm sorry about the full stop there, that was a mistake, ignore that, growth factor receptor binding protein 2, okay, and for short, that's GRB2. Now, GRB2 has a special domain in order to bind to the phosphotyrosine residue on the intracellular aspect of our receptor tyrosine kinase. Okay, so I'll colour this special domain in green here. Okay, and this special domain is known as an SH2 domain. So this, which I've coloured in green, this is a special domain within the structure of the growth factor receptor binding protein 2, and this is called an SH2 domain. Now, what does this stand for? This stands for Sark homology. Okay, so the S is for Sark, the H is for homology, and then the 2 is for 2. Okay, so Sark homology 2 domain. Okay, often people will read it instead as Sark homology domain 2 rather than Sark homology 2 domain. And the reason it's called that is it's a domain that's found in all sorts of different proteins. So all over the place in molecular biology you find Sark homology domains. Okay, and they were originally found in a protein that we will actually come across later called Sark, okay, which is a receptor, not a receptor tyrosine kinase, sorry, it's not a receptor tyrosine kinase, it's just a tyrosine kinase, okay, so it's one of these tyrosine kinase enzymes that's free within the cytoplasm. Okay, right. So, SH2 domains, therefore, bind to phosphotyrosine residues. That's what their special ability is, that they can bind to phosphotyrosine residues. Okay, so growth factor receptor binding protein 2 will bind to one of these phosphotyrosine residues on the intracellular aspect of our receptor tyrosine kinase dimer here via its SH2 domain. Okay, then it has another special domain that it's going to use to bind to the next protein along. Okay, so I'll colour this next special domain in red here. Okay, and this next special domain is a domain known as an SH3 domain. Okay, now this stands for Sark homology domain 3. Okay, so once again, it's a domain that you find in loads of different proteins, but it was first found in the Sark family of receptor tyrosine kinases. I'm sorry for continually saying that, of tyrosine kinases. Okay, right. So SH3 domain. Now, what's the special feature of SH3 domains? SH2 domains were special because they could bind to phosphotyrosine residues. What's special about SH3 domains? Well, the special feature of SH3 domains is that they bind to regions that are rich in proline residues. Okay, so they will bind to proteins which have a domain which has a lot of prolines in it. Okay, so let me just remind you of the structure of the amino acid proline. Okay, so once again I'll show it as though it's a residue. So here's the amino group. Now in proline you have a special R group, because the R group is going to actually involve the nitrogen in the amine group of the amino acid. Okay, so when you actually have a proline amino acid in a protein, there is not going to be a single hydrogen coming off this nitrogen because the other bond of the nitrogen is involved in the R group. Okay, so it's slightly unconventional for an amino acid. Okay, so after the amino group we then have the alpha carbon followed by the carboxylic acid group down here. Now, the R group then of the proline residue consists of 3 methylene groups that go round in a ring like so. Okay, so here's a methylene group, here's a methylene group, and here is another methylene group like so. And they form a 5-membered ring within the structure. Okay, so this is the structure of a proline residue then. Okay, so in proteins where you have a domain which is rich in prolines, known as a proline-rich region, that proline-rich region can bind to SH3 domains. So let me draw this here. So we've got our next protein here will have a special region which I'll colour in vivid purple here, which is going to be a region that's rich in prolines. So this first region here, this is going to be a proline-rich region. Okay, so it's going to contain a lot of these proline residues. Okay, now what is this protein that has the proline-rich region? Well, this is a protein known as SOS. Okay, now SOS stands for Son of Severnus, and I actually don't know the story of how it became known as that. Okay, it's like a name that you get out of developmental biology. Developmental biologists tend to come up with ridiculous names for things, and this looks like one of their sort of names. Okay, so SOS, it's basically a guanine nucleotide exchange factor for RAS monomeric G proteins. Okay, so before we discuss SOS, let's discuss RAS monomeric G proteins and then we'll come back to SOS. So at the moment we've had growth factor receptor binding protein 2 bind to a phosphotyrosine residue on the cytoplasmic tail of our receptor tyrosine kinase dimer. Okay, now we've got SOS binding onto the growth factor receptor binding protein 2. This will firstly recruit SOS to the underside of the phospholipid bilayer, and it will also activate it. It's now going to work on RAS monomeric G proteins. Okay, so I want to, before talking about SOS, talk about RAS monomeric G proteins. Okay, so RAS is a monomeric G protein. Okay, now these are similar and also different from heterotrimeric G proteins, which are more famous than monomeric G proteins. Okay, so the feature that all G proteins have is that they have 2 states. Okay, they have an on state in which they have guanosine triphosphate bound to them, and they have an off state in which they have guanosine diphosphate bound to them. Now, in monomeric G proteins, the G protein consists of just a single polypeptide, okay? And this polypeptide can either be in the on state where it has guanosine triphosphate bound to it, or it can be in the off state where it has guanosine diphosphate bound to it. So they are effectively on-off switches, okay? They are similar to the alpha subunit of heterotrimeric G proteins. In heterotrimeric G proteins, the alpha subunit can either have guanosine triphosphate bound to it, in which case it's in the on state, or it can have guanosine diphosphate bound to it, in which case it's in the off state. Okay, now in the off state, where it has guanosine diphosphate bound to it, it then also associates with two other different subunits known as the beta and the gamma subunits, which makes it a heterotrimeric G protein. But the principle of monomeric G proteins is actually simpler than heterotrimeric G proteins. Monomeric G proteins have this on state where they have guanosine triphosphate bound to them, and they have this off state where they have guanosine diphosphate bound to them. The difference between them and heterotrimeric G proteins is that the alpha subunit of heterotrimeric G proteins, when in the off state, then binds to a beta gamma subunit as well. Okay? But it's simpler in the case of monomeric G proteins because they don't do that. Okay, so there is a huge number of different monomeric G proteins. Okay, one family of monomeric G proteins is the RAS monomeric G proteins. Now, there are 3 genes for RAS monomeric G proteins. There is NRAS, okay, there is BRAS, okay, and there is also KRAS. Now, in the case of NRAS and BRAS, both of these only code for a single protein known as NRAS and BRAS. In the case of the KRAS gene, you have multiple splice variants. So let me just remind you of the concept of splice variants. Okay, so let these 2 parallel lines here, instead of representing the phospholipid bilayer now, let's let them represent a piece of double-stranded DNA. Okay, so let's now say that we have some gene here. So this region of this double-stranded DNA that I have now boxed, and I'm going to even colour it in, we're going to say that this is some gene. Okay, now one of the strands of this gene will be what's known as the coding strand. Okay, so I'll colour this in purple. So this strand that I've now coloured in purple there, this is the coding strand of the gene. And this is the strand basically that will be read by the RNA polymerase II enzyme to produce a piece of mRNA. Okay, so the enzyme RNA polymerase II, usually abbreviated to RNAP2, RNA for ribonucleic acid, the P for polymerase, okay, and then the fancy Roman numeral for 2. Okay, right, so RNA polymerase II is going to come along. Okay, it's going to work its way along the coding strand, and it's going to produce a piece of mRNA that's going to be complementary to that coding strand. So I will colour in RNA polymerase II here in blue. Okay, right. So RNA polymerase II works its way along the coding strand, synthesizing a piece of mRNA, okay, which is in blue here. Now, the other strand that RNA polymerase II does not use, just for nomenclature purposes, that's called the non-coding strand. Okay, so only one of the strands of DNA within a gene is used by the RNA polymerase II enzyme to actually make RNA. Okay, so we've got our piece of mRNA now. Here it is. Now, the question is this piece ready to go through a ribosome and be translated yet? And the answer is no. Okay, this piece of mRNA that we've just produced is what's known as a pre-mRNA, or it's also called a primary transcript, okay, or a primary mRNA transcript. Okay, so it is not ready yet to be translated. It firstly needs to go through post-transcriptional modifications. Okay, so this is the portion, the thing that we've just produced from the gene. Okay, so the reason it's not ready to be translated at the moment is that there are regions in this pre-mRNA that are not actually supposed to be translated. Okay, so some portions are supposed to be translated, and I'll box these now. So here's one, let's say this is another one here. Okay, and the portions of the pre-mRNA that are supposed to be translated are known as the exons. So these portions are the exons. So I'll highlight these in turquoise. So this portion here is an exon, this portion here is an exon, this portion here is an exon. The portions in between the exons, which are the portions that are not actually supposed to be translated, okay, so these 4 portions here. And of course, this is just a drawing. It will vary depending on your gene, how many exons you have and how many introns you have. So these portions that are in between the exons and which are not supposed to be translated, those are called the introns. Okay, so what has to now follow is a process called splicing, in which you cut out the introns that are in between the exons, and then you glue the exons back together, and what you end up with is a piece of mRNA that is much shorter than the original pre-mRNA, and this is known as a piece of mature mRNA. Okay, this piece of mature mRNA is then ready to go through a ribosome and be translated into protein. Now, in the case of certain genes, there is more than one way that you can splice the pre-mRNA. Okay, so you get your pre-mRNA here, and there are different ways you can splice it, and these different ways of splicing it will produce different pieces of mature mRNA, and these different pieces of mature mRNA will then code for different proteins, and these different proteins are then called the different splice variants of that gene. So you have one gene producing multiple different proteins. So let me give you a very nice, easy to understand example of this. So let's say that this exon right at the end here, this small one at the end, let's say that this is not compulsory, okay? And you do find this setup in certain pieces of pre-mRNA. Let's say that this exon here is optional, okay? So you can either include it or you don't have to include it. You can either have it in your piece of mature mRNA or you can cut it out as though it's an intron and throw it away, okay? That means that there are 2 different ways that you can splice this pre-mRNA. Either you can include all 3 exons and therefore get a big piece of mature mRNA, or you can just include the first and the second exon and get a smaller piece of mRNA, which I'll draw here. Okay? That means that there are 2 pieces of mature mRNA that you can produce from the same piece of pre-mRNA. Okay? And these 2 different mature mRNAs now will code for different proteins, and these different proteins came from the same gene, and they are therefore called the different splice variants of a gene. Okay, so it's not always that simple. Sometimes it's much more complicated than that. That's a really nice, easy example to explain and understand, so that's why I put that there. But the principle, the basic principle of different splice variants is that there are different ways of cutting up your mRNA and then restitching it back together, and therefore there are different mature mRNAs you can end up with, and therefore from those different mature mRNAs you can end up with different proteins, and those different proteins that all come from a single gene are called the different splice variants of that gene. So, for NRAS and BRAS, we only know one splice variant of each of those genes, and they're therefore called the NRAS protein and the BRAS protein. But for KRAS, there are 2 splice variants known, and I don't actually know if the reason for these 2 different splice variants are as simple as I've just explained here. Okay, it might be more complicated. So, in fact, it most likely will be. So the 2 splice variants then of KRAS are called KRAS 4A, okay, and then also KRAS 4B. So those are 2 different RAS proteins, but they're different in this quotation marks way. They're different splice variants that come from the same gene. So that leaves us overall with 4 different RAS proteins. Okay, now, for our purposes, all of these are going to do exactly the same thing. Okay, their involvement in cancer is slightly different. They are different proteins and they do slightly different things. For our purposes, it's good enough to say that they all do the same thing. So from now on, we won't talk about the 4 different RAS monomeric G proteins. We'll just call our monomeric G protein RAS, and when I say that, I mean it's one of these 4, but I don't no, neither do I care which one it is. Okay, so they all do the same thing as far as we're concerned. Now, where do the RAS monomeric G proteins live? Well, they live on the underside of the phospholipid bilayer. Okay, So let me draw my RAS monomeric G protein for the first time. So basically, these RAS monomeric G proteins, they are not integral membrane proteins. They are not sitting within the phospholipid bilayer, but they are attached to the underside of the phospholipid bilayer. Okay, now, as I said, it's a monomeric G protein, so it can be in two states. It can be in the off state, where it has guanosine diphosphate bound to it, and it doesn't then interact with its downstream targets, or it can be in the on state where it has guanosine triphosphate bound to it. Okay, at the start it's going to be in the off state, and it's going to have guanosine diphosphate bound to it, GDP here. Okay, in addition, it's attached to the underside of the phospholipid bilayer. How? How is the next question. Okay, so the reason it's attached to the underside of the phospholipid bilayer is that it has a lipid moiety stuck off the side of it. Okay, and this lipid moiety is called a farnesyl group, and I'll explain to you exactly what a farnesyl group is in a moment. Okay, so basically, the basic idea here is that we attach onto the side of our RAS protein an extremely long hydrophobic lipid group. Okay, this lipid group now does not interact at all well with water, so it means that the RAS protein isn't now going to be very stable in the cytoplasm, and instead what happens is this lipid moiety implants into the inner leaflet of the phospholipid bilayer so that it can interact nicely with the other hydrophobic molecules in that hydrophobic core of the phospholipid bilayer, and this hangs the RAS monomeric G protein off the bottom of the phospholipid bilayer here. Okay, so our RAS protein is going to be suspended onto the base of the phospholipid bilayer here via this farnesyl group that we have. Okay, so let me talk to you a little bit more about, not on that side, about farnesyl groups. Okay, so this is a common lipid modification that you can make to proteins. Okay, so farnesyl groups. Farnesyl groups consist of 3 molecules of isoprene polymerized together. Now, to understand what that means, you firstly need to know what isoprene is. So we're going to start off with what is isoprene. Okay, now people usually draw skeletal formulae for isoprene, but the skeletal formulae are kind of difficult to look at. Okay, so we're going to start off with a more blatant formula, which is molecular formulae. Okay, right, so isoprene molecules then. So, basically isoprene molecules have a backbone of 4 carbons, okay, which I'll put here. And they have 2 of these bonds being double bonds. So this bond here and this bond here are going to be double bonds. They then have a methyl group coming off one of these carbons up here, this one here, and a hydrogen coming off that one, and then hydrogen saturating everything else. Okay, so this is the structure of an isoprene molecule. Now, let's draw this in skeletal format, okay? So in skeletal format, we need to start by drawing 4 carbons. So remember, in skeletal formula, you don't show you don't show carbons. Okay, they're implicitly shown by the corners. You also don't show hydrogens coming off carbons. So here's our first carbon here. So let's draw the parallels here with colours. So this is our first carbon here in pink. Here's our second carbon in blue here. Okay. Here is our third carbon over here, okay, which I'll colour in green. So this one up here, this is our third carbon, and then our fourth carbon is down here, and this one we'll have in red. Okay, now, between our first and our second and our third and our fourth carbons, we need double bonds. So let's promote these up to being double bonds here, like so. Okay, and then of our third carbon, we need a methyl group. Now, in skeletal formula, you just show a methyl group literally as a stick, sticking up like that. Okay, so that's that one. There is supposedly a difference between that colour and that colour. Okay, you can just see it. And that then is the finished isoprene molecule in skeletal format. And I'll just redraw it out here again. Okay, and that's why I didn't think it was a good idea just to draw this first time, because that, you look at that and it doesn't really leap to your mind what that means, whereas this is far more blatant. Okay, right, so this is an isoprene molecule. Now, what you can do is you can polymerize isoprene molecules together. Now, let me explain how this is going to work. Basically, what you're going to do is you're going to break the second of the two bonds between this first and this second carbon here. So you're going to break this bond here in blue. Okay, you're also going to break the second of the two bonds between the third and the fourth carbon over here. So we're going to break this bond in blue over here as well. Imagine sending one electron from this bond back to this carbon and one electron from this bond back to this carbon. So remember, in covalent bonds you have two electrons, one from each of the two members. Imagine sending the electron in this bond from this carbon back to this carbon and the electron in this bond from this carbon back to this carbon. Okay? And do the same for these two carbons here. So send one electron here, one electron here, one electron here, one electron here. That's going to leave all of these four carbons with a single free electron. This carbon and this carbon will now form a new bond between themselves. They'll form a double bond. Okay, with their two free electrons. So this carbon will put an electron in, this carbon will put an electron in. That then leaves the first and the fourth carbons here with free electrons, which I'll show like this. Okay, what we can then do is bring along another isoprene molecule that has done this exact same thing. Okay, so it's broken this bond and forged a new bond here. That will then have free electrons on the first and the fourth carbons. And you can bind this carbon here to this carbon here via a single bond. And of course you can continue this on. You can bring another isoprene molecule here, another one here, another one here, and that's how you can polymerize isoprene molecules together. Now we're not going to go very far, we're only going to polymerize 3 of the things together, so we're almost done in fact. Okay, let's just bring in another one here. Okay, and then we're done. We add this one onto this one. Okay, so there was a free electron here and there was a free electron here. Okay, that only leaves this carbon and this carbon now with free electrons. Okay, so what we're going to do is we're going to attach this farnesyl group onto a cysteine residue within the polypeptide of the RAS protein. So let me show you how. Okay, so once again, I haven't actually stressed this as yet, but what I'm going to show you is not a molecular flow diagram. It's not an electronic flow diagram. I'm not showing you how these reactions occur electron by electron. I'm not showing you the organic chemistry of it. I'm just telling you what overall happens so that you can understand what has happened rather than what actually happens. Okay, so let's take our cysteine residue here. Okay, and we're going to attach onto the cysteine residue one of these farnesyl groups. So firstly, let's just have a look at a cysteine residue. So a cysteine residue's R group consists of a methylene group with a thiol group coming off it. Okay, so this is the structure of cysteine. Now, this group that we have here with a sulfur atom and a hydrogen coming off it, this is called a thiol group. Okay, and it has very similar chemical properties to an alcohol group. And the reason is that if that sulfur was an oxygen, that would be an alcohol group. Now, sulfur is in the same group of the periodic table as oxygen, so has very similar chemical properties to oxygen, and therefore these thiol groups have very similar properties to oxygen. And what we're now going to do is we're going to break this bond between the sulfur and the hydrogen atom there. Okay, and once again there are 2 electrons in that bond. You're going to send 1 electron back to the sulfur atom and 1 back to the hydrogen atom. Okay, and again this is not an electronic flow diagram, this is how you can make sense of what's going to happen. How you can see that what's on one side adds up to what's on the other side. Okay, so imagine sending 1 electron back to this sulfur and 1 back to this hydrogen. Then you're going to bring this thing in and insert it in between them, basically. So you're going to bind the sulfur, which now has a free electron from the breaking of this bond, to this carbon, which has a free electron here. Okay, so you're going to form a bond, and this is this first carbon now. Okay, in blue here, it's going to attach there. Okay, bound to this sulfur atom. Now continue the structure on. Okay, so just copy this out. So here's our first isoprene molecule. Now we're on to our second up here. Okay, there's our second. And here then is our third, and it might help you to draw them one at a time and then connect them up. Okay, like so. And now this carbon, which has got a free electron, we're going to bind the hydrogen here onto that. Okay, and we'll now have this farnesyl group linked off our cysteine residue. That's just showing you how this makes chemical sense. It's not showing you actually how the cell does it. There'll be a horrendously long pathway for how the cell does it, which I don't know. But that's the principle, that you're attaching these farnesyl groups onto cysteine residues. Okay, right. So every RAS protein gets one of these farnesyl groups. So this group where we have 3 isoprene molecules linked together, this is called a farnesyl group. So the entire thing that we've now added onto the side of our cysteine residue. Okay, so all of this that I'm circling in green here, this is a great farnesyl group. Okay, and just one other little bit of terminology. This bond that you have here, okay, where you have a carbon atom bound to a sulfur atom, which is then bound now to another carbon atom. There's a name for that, okay, that's called a thioether link, okay. So if that was an oxygen atom sitting where the sulfur atom is sitting, it would just be called an ether link. So it's very similar to an ether link, but just to show us that we're dealing with a sulfur atom rather than an oxygen atom, it's called a thioether link. Okay, so all RAS proteins are going to get one of these farnesyl groups added onto one of the cysteine residues within their polypeptide. Now, usually it's the final cysteine residue within their polypeptide. Okay, and this is going to implant into the inner leaflet of the phospholipid bilayer, okay? And the reason is that this is extremely hydrophobic, this chain here. All of these carbons are just bound to other carbons and bound then to hydrogens. All of those bonds are non-polar, so they're not going to interact at all well with water, okay? So this implants into the inner leaflet of the phospholipid bilayer and holds the RAS monomeric G protein at the inner leaflet of the phospholipid bilayer. Okay, so we'll call it there for this video, and in the next video we'll come back to Son of Severness, and we'll see how it's going to catalyse the activation of the monomeric RAS G protein.